Hello students, I am teacher Svetlana, and in our today's lecture we will continue to learn the components of a eukaryotic cells, which are ribosomes, nucleus and cytoskeleton, which is covered in the chapter Cell Structure and Organization. So let's begin. Ribosomes. You are aware that ribosomes are protein factories of the cell. They use the genetic information to synthesize proteins. Ribosomes were first observed as dense particles in electron micrograph of a cell by scientist Balade in 1953. Ribosomes are made up of ribosomal RNA, which is also called as rRNA, and proteins. They do not have any membranous covering around them. In eukaryotic cell, the ribosomes are present in mitochondria, plastids, and in the cytosol. Ribosomes in the cytoplasm are either found attached to the outer surface of the rough endoplasmic reticulum and the nuclear membrane or freely suspended in the cytoplasm. Both are similar in structure and are ATS type. Each ribosome is made up of two subunits, a larger subunit and a smaller subunit. Have a look at this figure here. These are the two types of ribosome. One is the prokaryotic ribosome and another one is the eukaryotic ribosome. You have two subunits which come together to form the entire ribosome. Bound ribosomes generally produce proteins that are transported outside the cell after processing in the endoplasmic reticulum and the Golgi complex. Example, bound ribosomes of Eckner cells of the pancreas produce pancreatic digestive enzymes. So, bound ribosomes, they generally produce proteins that are transported outside the cell. Free ribosomes come together and form chains called as polyribosomes for protein synthesis. Free ribosomes generally produce enzymatic proteins that are used up in the cytoplasm like enzymes required for the breakdown of sugars. So students, when we are talking about ribosomes, multiple ribosomes come together and they form a unit called as polyribosomes. Now you will come across this term when you will study the process of translation, that is the synthesis of proteins, that is how genetic information is used for the production of or formation of certain proteins in our body. At that time, ribosomes play a very important role. Both types of these ribosomes can interchange positions and functions. Number of ribosomes is high in cells actively engaged in protein synthesis. It's obvious that since they play a very important role in the synthesis of proteins, so the cells which are actively engaged in protein synthesis, the number of ribosomes is usually high in such cells. You can see in the prokaryote we have the 70S type, that is a smaller 30S subunit and a larger 50S subunit comes together, and in the eukaryotic cell it is of the ATS type, where you have the smaller subunit which is 40S and the larger subunit which is 60S. So this is ribosomes. The most important organelle in a eukaryotic cell is the nucleus. Structure of the nucleus of the eukaryotic cell becomes distinct in a non-dividing cell or during interphase. Such an interphase nucleus is made up of nuclear envelope, nucleoplasm, nucleolus and the chromatin network. So you can see here is the image of the nucleus. This is how the nucleus appears at a time of interphase, that is when it is not undergoing any cell division, and when the cell starts undergoing cell division, at every stage the shape or the appearance of the nucleus starts differing. So in an interphase cell, when the cell is not dividing, the nucleus shows these following components. Let us have a look at each of these components. The first one is the nuclear envelope. This outer layer is your nuclear envelope. It is a double walled delimiting membrane of the nucleus. Two membranes are separated from each other by perinuclear space, which is around 10 to 50 nanometers. Outer membrane is connected with endoplasmic reticulum at places. So the outer membrane that is here present, it has at places it is attached from the outside. You have the endoplasmic reticulum which is in close proximity or attached to the outer membrane. It also harbors ribosomes on it. The inner membrane is lined by nuclear lamina, a network of protein fibers that help in maintaining shape of the nucleus. The two membranes along with the perinuclear space helps in separating the nucleoplasm from the cytoplasm. So you have these two membranes, the outer membrane and the inner membrane. Now the inner membrane is surrounding the nucleoplasm which is present inside the nucleus, and the outer one separates the inner contents of the nucleus from the remaining cytoplasm. However, nuclear membrane is not continuous. At places there are small openings called as nucleopores. These are tiny nucleopores that are present in it. These nucleopores are guarded by pore complexes which regulate flow of substances from the nucleus to the cytoplasm and in the reverse direction. Here students, in this image you can clearly see the endoplasmic reticulum which is coming in contact with the outer membrane, and it has numerous ribosomes attached to it. So here in this case it forms the rough endoplasmic reticulum. Nucleoplasm or the karyolymph. It consists of various substances like nucleic acids, protein molecules, minerals and salts. It contains chromatin network and the nucleolus. This is the nucleolus, and the thread-like or cord-like structure is called as the chromatin. Nucleolus is another component which is not bound by a cell membrane. There is no cell membrane to this organelle. What does this nucleolus do? It is made up of rRNA and ribosomal proteins, and it is best known as a site for ribosome biogenesis. So this place is a site where ribosomes are formed. Depending on the synthetic activity of a cell, there are one or more nucleoli present in the nucleoplasm. For example, cells of the oocyte contain large nucleolus, whereas sperm cell contains a small inconspicuous one. They appear as dense spherical bodies present near the chromatin network. So here you can see it's slightly dense as compared to the chromatin network. They produce RNA and ribosomal proteins which are then transported to the cytoplasm and are assembled together to form the ribosome. So it forms the ribosomal proteins which move out and into the cytoplasm where they get assembled into the ribosomes. Then we have the chromatin material. This pink color thread-like structure is our chromatin material. Nucleus contains genetic information in the form of chromosomes, which are nothing but DNA molecules associated with proteins. In a non-dividing cell, the chromosomes appear as thread-like network and cannot be identified individually. This network is called as the chromatin material. The chromatin material contains DNA, histone and non-histone proteins and the RNA. In some region of the chromatin, DNA is more and is genetically active, and this region is called as euchromatin, and some regions that contain more proteins and less DNA and they are genetically inert, that is they are genetically not active, that region is called as heterochromatin. When the cell prepares to divide, the chromosome coils and gets condensed. At metaphase stage, they become distinct and can be clearly identified. You will study this process as well as the structure and the types of chromosomes in the next chapter or a later chapter. Every species of the living organism has specific number of chromosomes. Like for human cells, we have 46 chromosomes which are grouped together in 23 pairs. The nucleus contains entire genetic information, hence plays an important role in heredity and variation. It is a site for synthesis of DNA, RNA and ribosomes. It plays an important role in protein synthesis. Chromosome number being constant for a species, it is important in the phylogenetic studies. Nucleus thus is the master cell organelle. The component of a eukaryotic cell is the cytoskeleton. With advancement in light and electron microscopy, scientists revealed the presence of network of fibrins throughout the cytoplasm. It is called as cytoskeleton. Cytoskeleton consists of microtubules, microfilaments and intermediate filaments. You can have a look at this figure here. You can see all these filaments. These are your intermediate filaments. These are the microtubulins, and these are the microfilaments. Microtubulins are made up of protein tubulin, microfilaments are made up of actin, and intermediate filaments are composed of fibrous proteins. Cytoskeleton helps in maintenance of the shape of the cell, contraction of the cell, mobility of the cell, and cell organelles, changes in the shape of the cell, and cell division. So basically it forms a structure where the organelles are suspended. It also gives flexibility to the cell. It helps in contraction, helps in the mobility, movement of the cell and the cell organelles. Many eukaryotic cells also show presence of cilia and flagella. We know that these are used in locomotion. They are fine hair-like membrane-bound protoplasmic outgrowths that occur on the free surface of the cell. They generate a current in the fluid medium for passage of material and locomotion. Cilia are small in size and many in number. Cilia act as oars causing movement of the cell. Flagella, on the other hand, is longer and few in numbers. Flagella present in prokaryotic bacterium are structurally different from that of the eukaryotic flagella. Cilia or the flagellum consist of basal body, basal plate, and a shaft. This image here gives you an overview of how the structure of the eukaryotic cilia or the flagella looks. Basal body is placed in the outer part of the cytoplasm. It is derived from the centriole. It has 9 peripherally triplets of fibrins. Shafts is exposed part of the cilia or the flagella. It consists of 2 parts, sheath and the axonema. Sheath is a covering membrane of the cilium or the flagellum. Core, called as axonema, possesses 11 fibrils running parallel to a long axis. It shows 9 peripheral doublets and 2 single central fibrins, that is 9 plus 2. This here, if you count, you will get this rings, microtubulin rings. The central tubes are enclosed by the central sheath. The sheath is connected to one of the tubulins of the peripheral doublets by a radial spoke. So you have these radial spokes that are there, which bring about the connection of the tubulins to the sheath. Central tubulins are connected to each other by bridges. The peripheral doublets are connected to each other through linker or interdoublet bridges. So here in between also they are interconnected with the help of the linker or interdoublet bridges. Centrioles and centrosomes. Centrosome is usually found near the nucleus of the animal cell. It contains a pair of cylindrical structures called as centrioles. The cylinders are perpendicular to each other and are surrounded by amorphous substance called as pericentriolar material. Each cylinder of the centriole is made up of 9 sets of the triplet microtubulins made up of tubulin. So here, if you have a look, this is the centriole, which is having this triplet microtubulin, and these microtubulins are interconnected by a non-tubular material. Evenly spaced triplets are connected to each other by means of non-tubular proteins. At the proximal end of the centriole, that is at one end, there is a set of tubulins called as hub. The peripheral tubulins are connected to the hub by means of a radial spoke. So here you can see that they are interconnected, and this is your radial spoke which is connecting the triplet microtubulin. Due to this, the proximal end of the centriole looks like the cartwheel. So at the end, if you see from one end, it appears like a cartwheel. The centrosomes help in assembly of the spindle apparatus during cell division. It forms the basal body of cilia and flagella. So if you go through this data that is given below the diagram, it will help you to understand its features. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.